Okay, so, and we'll talk about this in lab too, but this is about DNA and cells. So the human cell has two main kinds of DNA. So one is nuclear, which is found in the nucleus, and it's genomic DNA. And the other is mitochondrial. And mitochondrial DNA is only found in the mitochondria. Okay, so nuclear DNA is a big molecule, and it's a polymer. Okay, so it's a polymer, and it's made up of a whole bunch of repeating units, and the repeating units are called nucleotides. So nucleotides are made up of a sugar, a phosphorus molecule, a phosphate, and a nitrogen-containing molecule that's a base. And I'll show you in a picture here. Okay, now there's 4 types of bases found in DNA. So there's adenine, guanine, cytosine, and thymine. So A, G, C, and T. Okay, now this picture is from the book, and it shows the sugar molecule, and the sugar molecule is attached to the phosphate. And from there, then we have the nitrogenous base. So the nitrogenous base is what makes it the A, C, T, or G. But the whole unit is a nucleotide. So it's a sugar, a phosphate, and then these nitrogen bases. The way that the molecules are organized is they're put together. The way that they're linked together is it's sugar, phosphate, sugar, phosphate, sugar. So they call it a phosphodiester backbone. Okay, so I don't know that that's in your book, but so phosphodiester backbone. So those are bonds that are linking it together. So if you think of it kind of as a ladder, the nucleotides are going to be the steps on the ladder, but the sugar and the phosphate are going to be like the arms of the ladder. Okay, so nuclear DNA it's a double helix. So Watson and Crick and Franklin figured that out a while ago, I guess in the 70s, 60s or 70s. Okay, now all of the nucleotides, they pair very systematically. Okay, so adenine always pairs with thymine. So A pairs with T, and those are straight-edged letters. So straight goes with straight. And guanine always pairs with cytosine. So C pairs with G, and those are the curvy letters. Okay, so the book has a picture of the double helix. So and we have a model I'll show you in lab, but C is always going to pair with G. And that makes up the steps of the ladder, or the rungs of the ladder. And then the sugar and the phosphates, they're going to make up the backbone of the ladder. So if you have a ladder and you twist it, that's a double helix. And that's what the DNA, that's what our chromosomes have. Okay, so DNA before our cells divide. So our cells have to divide when we get injured, when we grow, just to maintain our body. Okay, so in order for our cells to divide, the DNA has to duplicate itself, and that's called DNA replication. There are a lot of different enzymes involved in DNA replication. So what happens is the DNA is going to unwind the double helix, and then we have nucleotides in our body that are going to be put together using enzymes. I'll show you a video. Okay, so the double-stranded helix unwinds, and then some other enzymes are going to come in, and that's how we're going to form 2 new double helixes. It's called semi-conservative replication because we're hanging on to an old strand and we're making a new strand off of the old strand. Okay, now one of the interesting techniques that is used in forensics and paternity testing and diagnosing diseases, all sorts of things, is called polymerase chain reaction, or PCR. So PCR has become really common. I think it was maybe discovered in the 80s. The guy that discovered it actually got a Nobel Prize for it. Okay, so PCR stands for polymerase chain reaction. So it's a chain reaction. Polymerase is an enzyme that's going to build things. Okay, so remember, anytime, and I don't know if you guys ever learned this, but Anytime you see something with the ASE on the end of it, that is like the universal symbol for enzyme, okay, if it ends in ACE. So polymerase is an enzyme that builds things, builds polymers, okay? And then chain reaction means it's just going to keep building, kind of like knocking down the dominoes. Okay, so it's a technique for, what we're going to do is we're going to copy small pieces of DNA or broken pieces of DNA that could be found at a crime scene, and they're not necessarily in a cell. So what it's going to do is it's going to allow us to amplify little teeny tiny quantities of DNA, and make enough DNA that we can actually use it and do something with it. Okay, so we'll go over the steps of PCR in lab because we're going to do it. So what happens first is the DNA is going to be heated and it's going to separate the double helix. Then what we do is we take these things called primers. And primers are these little short pieces of DNA, and they're made, so we make this DNA to target specific regions that we want to amplify. So because of the A's, T's, C's, and G's, and the way that they bind, this complementary base pairing, we can make pieces of DNA that will bind with target DNA. So we can figure out what they need to be. We can pay like $1 a base, maybe it's not even that much anymore, and have it produced. Okay, then what'll come up next is we'll use an enzyme which is called DNA polymerase, and we can use it and we can add A's, T's, C's, and G's to it, and then we can build new DNA molecules. So basically what we do is we heat, we add the primers, and then we build it out. So I'll go over all this with you in labs to make sure that you understand. We repeat it like 20 to 25 to 30 times. It takes about 2 hours, and we can copy millions of pieces of DNA. Okay, I don't necessarily like this book, but look like the book's picture, but it has target DNA. Okay, and then it has primers. Okay, so the primers are going to be complementary to the target DNA. So A is going to pair with T, C is going to pair with G, and they're going to flank a sequence of DNA that we're interested in copying. Okay, so we want to copy this stuff that's in the middle. So we're going to make DNA that we know will bind to the edges of it. And those are called primers. Okay, now, forensics uses the idea of PCR, so this technique of PCR, and then this concept of short tandem repeats, which are called STRs, to identify individuals. Okay, so short tandem repeats are places on the chromosome that have information that's repeated over and over and over and over again. Okay, so they're usually 3 to 7 base pairs in length. Okay, so 3 to 7 A's, T's, C's, and G's, but they're repeated over and over and over again. Okay, so they're not coding for a gene, so they're not coding for anything that could be used for like health purposes, insurance, or something like that. But they're coding, but they're different among individuals. We'll also do this in lab. Okay, so STRs are very short, so they're less than 450 base pairs long. So because it's a short piece of DNA, if the DNA is old and it's starting to break down because it hasn't been like stored in the freezer, okay, you could recover some short pieces and you could amplify them and use STRs to try to identify an individual. Okay, so we're going to use PCR to amplify STRs. STRs usually have a 2-band pattern, so I have to show you what that means in lab. It makes it really straightforward to interpret it. If you have, I'll have to show you in lab the banding pattern and peaks and stuff like that. So if you have 2 individuals, instead of having just 2 Okay, so one person will give 2 bands, but if you have 2 individuals, you'll end up with 4 bands. So I'll try to explain that to you in lab. Okay, so there is a commonly used SCR, it's TH01. Okay, so TH01 has a repeating sequence, it's AATG. Okay, so AATG. So different people have different numbers of AATG on their chromosome. Okay, so 7 TH01 variants have been identified. Okay, so they have between 5 to 11 repeats of that. So somebody could have AATG 5 times. 1, 2, 3, 4, 5. Somebody else might have it 11 times. Some people have it 6 times, and other people have it 8 times. Okay, now, usually what happens is each person, because we're getting a chromosome from our mom and a chromosome from our dad, we have 2 STR types for this TH01. So we get one from each parent. So my dad might have 6 and my mom might have 8. Okay, so they're saying the DNA type known as TH0168 is found in approximately 3.5% of the population. Okay, so I would be that 3.5%. So because I got a 6 from my mom and an 8 from my dad. Okay, this is supposed to be a picture of that. So here's 1, 2, 3, 4, 5, 6. So 6 copies of AATG on one chromosome and 8 on the other. Okay, so that's what it would look like in the DNA. Now, that's just one STR. Okay, so it's only one. You couldn't, if 3.5% of the population has that type, you wouldn't be able to convict someone or identify an individual. So you'd have more like class data, a class characteristic. Okay, now, there's hundreds of different types of STRs in the human genome. Okay, the more that we can characterize, then that means we can use the product rule, and the smaller will be the percentage of the population which a particular combination of STRs can emanate, okay, or come from. Okay, so you can look at many STRs and reduce the chance, or like narrow down your suspect pool. Okay, now you can multiplex. Okay, so when you're doing PCR, you don't have to do one primer set at a time. So you could do 10 at a time, and you can run that data, and you can get lots of data really fast. Here's a triplex, okay, or triplex. So there's 3 STRs. So we have a question stain, and we have the control stain. And when we run these on a gel, what we're going to do, I have to explain all that to you. You're going to get size markers. So you'll have a control on here. So here's what FGA looks like. So they got 2 bands for FGA. This person had 2 bands for WVA, and then 2 bands for this D3S1358. Okay, now the control, okay, so we could say this came from, maybe this came from the victim. Okay, they match. So they are the same sizes on the gel. So they look identical. And that's 3 STRs. That's still not enough to say for sure that it's that person. Okay, 3 is not enough. Okay, there are 13 STRs that are commonly recorded in CODIS. Okay, so if you get 13, they say it's a 1 in a billion chance that it's that person. Okay, so this all 13 matching is good. Okay, so because STRs occur independently of each other, the probability of biological evidence having a particular combination of STR types is determined by the product of their frequency of occurrence in a population. So it's the product rule again. So you can figure out what's the odds of somebody having THO1, that pattern. What's the probability of someone having the WVA1, okay? And then you multiply them all together, and that gives you the probability that that could be your person. Okay, so currently 13 U.S. crime labs have standardized 13 STRs. So that's the goal, is to have all 13 match. And they're kept in CODIS. I'll show you a picture. Okay, so with STR, as little as 125 picograms of DNA is required. So a pico is really, really, really small. So a picogram is smaller than a nanogram. Okay, and I think a nanogram's small. A nanogram is like 10 to the negative 9th. So a picogram must be 10 to the negative 12th, or 10 to the negative, let's see. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. 10 to the negative 12th grams. Okay, so 12 zeros. Okay. It only would take 18 cells. 18 cells with DNA. So you could have as small as 18 cells, which is like so tiny. It wouldn't even, I mean, wouldn't fill the head of a pin. So it's crazy. So it could be something that you touched. They could get DNA from that. Okay, now in lab, I will show you how to do gel electrophoresis, and we're just going to use agarose gels. Nowadays, there's a much more sophisticated way to do it, and we don't have that here, but it's called capillary electrophoresis, and what it does, instead of giving you a banding pattern, it gives you peaks. So the peaks kind of make it, I guess, a little bit easier to match things up. So we can talk about this in lab. It's faster, it's cheaper, it's probably more accurate because it's probably more digitized than what we do. Okay, now you can determine sex of the DNA. So there's a gene, it's called I'm not actually sure, I never pronounce it's amelogenin. So I think it's amelogenin. So it's a gene for tooth pulp, but on the X chromosome, it's shorter than it is on the Y chromosome. So remember, guys are XY and females are XX. So if you're a guy and we're looking for this I can't say it right now, the amelogenin gene, okay, they have 2 bands. Guys would have 2 bands. Females would just have 1 band. So that's one way to figure out sex. Okay, another way to figure out sex is they have STRs that are only found on the Y chromosomes. Okay, so if the guys are XY, they have STRs, so short tandem repeats, that are only found on Ys. So that means if there's more than one male involved in the crime, so like it's a gang rape, okay, and they want to see what men are involved in it, they can look at the Y STRs and determine how many different guys were there and maybe identify it to individual. Okay, now mitochondrial DNA is DNA that's found in this little tiny organelle that's called the mitochondria. Okay, the mitochondria, they have their own DNA, and what they do is they make energy for the cell, make energy for our body. Okay, so they call it mtDNA. It's always inherited from your mom. Okay, so and I'll explain that in lab, but if you think about an egg and a sperm, okay, sperm are pretty much just DNA in there. They don't have any organelles, whereas the mom has the egg, and the egg has all the cell components in there. So the sperm fertilizes the egg, and then the offspring is going to inherit all the organelles from the mom. So everything comes from the mom, organelle-wise, and mitochondrial DNA. So mitochondrial DNA isn't as good as STR analysis from nuclear DNA. However, if there's like a burned body or the nuclear DNA is broken down, they can use mitochondrial DNA to try to narrow things down. It's good for hair analysis. In the book, they talk about the Vietnam unknown soldier grave. They couldn't, I guess because it was so degraded, the body was so degraded. But however, they were able to exhume the corpse. And there were like, I guess, 7 or 8 different families that thought it was their relative in that grave. So they were able to look at the mitochondrial DNA and narrow it down to one family. So they did identify it. It's more rigorous. Okay, there's a smaller amount of it. It costs more. It's more time consuming. If you don't have to use it, you don't want to use it. And if they all had the same mom, if you all had the same mom, you wouldn't be able to tell the difference between people. There are 2 regions in mitochondrial DNA that they target. Because they're very different. So they're called HV1 and HV2. Those are highly variable or hypervariable, region 1 and region 2. So they'll look at those places because they're very different, and they can tell differences between families. Okay, CODIS is a combined DNA index system. So it's developed by FBI. It maintains local, state, and national database DNA profiles, convicted offenders, unsolved crime scene evidence, missing people. Right now they're saying 470,000 profiles from unsolved cases. 13 STRs is what they use. Let's see, the slide to turn. Okay, if you're going to collect evidence for DNA, you're looking for blood, semen, saliva, skin cells, so touch DNA, so something that somebody touched, like an object, like maybe a cup, a glass, cigarette, tape, a bullet, fingernail. Okay, could be hair, could be bone. So it's definitely, DNA is very powerful. Okay, so you can use bottles, cans, glasses, cigarettes, bite marks, envelopes, all those things, toothpick, all those things to collect DNA. Maybe a mask, okay, a hat. Okay, so this is kind of redundant, but you have to collect the clothing with blood evidence from victim and the suspect. If you want to get dried blood off, you use a sterile cotton swab that's moistened with distilled water, and then you swab it. Then you air dry it, and then you're going to put it in either a swab box or an envelope. And then you have to make sure that you have the standard and reference specimens in order to proceed, because you only are as good as your standards.